Okay, chapter 3, cell structure and function. Any idea why there would be a chapter on cells in human anatomy and physiology course? So we had chemicals, we had chemistry, right? Chapter 2 is over chemistry. Chapter 3 is over the cell. Any idea why? Because it's what? Right, so the smallest unit of life, basic unit of life, you'll hear all of those. And so in order to understand how the human body works, so get to understand how the cells work, right? Because what's actually doing the work of the body? Exactly. So when you think of your circulatory system and how the circulatory system is important in sustaining life, well, every part of the circulatory system is made of cells, right? So those cells have to be performing some particular job. For example, the heart. So the heart is made up of cells, okay? And we get into the different tissues in organs, but if it were not for those cells doing their job, what would happen to the heart? It would stop, okay? So we're going to look at the important structure and functions of cells, and it's probably the last time you'll hear me say that this is a review of Biology 103. A lot of chapters 2 and 3 are just a review of Biology 103 for those of you who took 103. So I try not to spend too much time on this, just trying to go into a deeper area so you all can really see what's going on with our cells. So a few things about cells. The modern cell theory states, for one, that all living things are what? Composed of cells. So we know that the whole universe is made up of atoms, right? But everything considered living is made up of atoms, which make up cells. Okay, so cells are the basic structural and functional unit of life. And all living things are made up of cells. Okay, smallest structural and functional unit of life. Make sense of that. The smallest structural and functional unit of life. So, can you make a distinction between the meaning of saying the cell is the basic structural unit of life versus saying it's the smallest functional unit of life? What's the difference in the two? Right. So structure, as she said, will be just physically what it's made of. Right? So the smallest structural unit of life means that anything living, the basic part of that is going to be the cell. Okay? Anything living, the basic part of whatever that living thing is the cell. And then the functional unit, how it works. So whatever is living, the function of that living thing is also starting at the cellular level. So it's what the cell actually does that allows the individual living thing, which is called an organism, to maintain life. An organism's structure and functions are due to the activities of its cells. So again, if you look at living things as cells, the function of the living thing is going to be tied into the function of each individual cell and the activities of those cells. Cells come only from division of pre-existing cells, not from living matter. And that's also an important one. because I know a lot of times people will hear things outside that, for example, I remember someone telling me that they didn't eat pork because pork turns into worms. Yeah. So pork is evil. If you would just let it sit out, it won't be pork anymore. It'll be worms of some sort. So in order for a living cell to be created, it comes from a pre-existing living cell. And what is that about? Have you all heard people say things like that? Well, yeah, pre-existing cells come from cell division, from a prior cell. But what my question is, have you all ever heard people say that non-living matter can become living, can turn into living? This says that's not possible. And what this young man was talking about was what people used to think, maybe in the 17th century, that if you put a piece of meat out, you'll see maggots on it. So that means the meat turned to maggots. But someone way back then did a study. They put the meat out, covered it, and there were no maggots on it. Versus the piece they had out exposed, there were maggots on it. So that tells you something else happened, right? Which is what? Flies, right. So if it's open and exposed, flies will land on it and lay those eggs. Okay, so that is an important one that I like to bring up. Cells come only from pre-existing cells, not from non-living matter. Cells of all species have many fundamental similarities in their chemical composition and metabolic mechanisms. So some of the things common to all living things are their cells, and the cells themselves are very similar across species. Okay, cells are the building blocks of all plants and animals and all other living things as well. And so where does a new individual come from? So thinking in terms of humans, where did we all start off? Right, so we started off as, what is that called? When the sperm fertilizes an egg? Zygote. So we all started off as one cell, right, by the fertilization of an egg by a sperm, and then that one cell just keeps duplicating, and they all duplicate, and you eventually get a full human being. So, and you all remember mitosis. When mitosis occurs, the cells are just duplicated, but they're going to be identical to the original cell, the mother cell. Okay? But there's something else interesting. So if you have one cell and this cell supposedly divides, when it divides, there are two cells that are identical to each other. Right? That's mitosis. And then those cells keep dividing. So how do we have, first of all, think about the organs in our body. Just pick two organs.
The heart and lungs.
The heart and the lungs. Or heart and the brain. Let's take those two. The heart and the brain don't look anything alike, do they? But they're both made up of cells. The same cells that came from that original zygote. Some of the cells, what happens is initially they are all identical to each other. But there's something that happens that tells some of those cells to start doing something different. Start becoming different in your appearance and your structure and your function. So when you look at individual cells of the brain versus individual cells of the heart, they look nothing alike. Okay? There's the picture here. So these are cells of the nervous system, the brain cells that send all these signals. They're specialized for what their job is. Muscle cells are made up of or muscle tissue is made up of cells like these. So the cells themselves appear different, but what is it that makes them start acting different from each other? DNA. That's interesting. But every single cell in our body has the same exact DNA. So how are they different? RNA is the same also. So the instructions are the same. So DNA, which we'll get into, is the instructions for the cells. But something at some point tells this group of cells to go to the brain area, go to the heart area, and the cells start becoming cells that are going to have a particular function. Think about muscles. What do we know muscles do? Just off the top of your head. Muscles contract, but what purpose do they serve in our body? Movement, right? So just think of any movement you intend to make, chewing, turning your head, picking up a pencil. In order for you to do that, your muscle cells have to contract, as someone said. And contraction just means shorten. So if you have so just think of a pencil. If the pencil was to contract at a particular time, it just gets shorter. Okay? So if that's this muscle here, my biceps brachii, when it gets shorter, whatever's connected to is pulled. Okay? So the grand plan was in order for movement to occur in humans, there has to be some way to cause movement of the limbs. So these cells have to be long, right? If you have a short cell, contraction won't do a whole lot, right? Does that make sense? So muscles structure is based on what its function is. Muscle cells are very long, so that when they contract, there's some actual movement that occurs. Okay, so that's the function of the muscles in our body. Heart muscle cells are a little shorter, but they don't have to be as long, and we'll get to that at some point. But look at neurons, nervous tissue. Those look completely different from any other cell we have in our body. And what do you know the nervous system's job to be? Really so many. It's just something basic. Good. Negative feedback. So if you all think back to the components of negative feedback, you have what? What's the first part from chapter 1? Receptor, right? And then what? Control center. Okay, so stop right there. How does a signal get from a receptor, let's just say on my finger, to my brain? There you go. So neurons are special because they have to be able to send a signal from my fingertip, and somehow it all gets all the way to my brain. So neurons are built in a way that they're able to transmit signals great distances. So they're not long completely, but they have long extensions. And those cells are able to transmit electrical impulses, unlike the other cells we have. Yes?
Well, all of them be close together like that, or are they like pretty much balanced?
What do you mean? Well, the way the tissues are made, they are going to be in a particular area. And this is all during the process of development, early in development. At some point, they'll migrate to a particular area. And it is the DNA. But what we're going to see later is that the DNA of certain cells tell that cell to make certain types of proteins. Yeah. Okay, so that's what makes all of our tissues different. Just want you all to look at it in a little bit deeper than you probably already have in the past. Just think of the body is made up of all these cells. The cells are pretty much the same because they all have a nucleus, they all have mitochondria, ribosomes. So they're alike in those terms, but their eventual shape, structure, and function will have to do with what their job is. So 200 different types of human cells. The types differ in size, shape, subcellular components, and functions. So all of the different cells we have, their shape and function is based on what their job is for the body. All right, so what is it called? That word, differentiation. Cytoplasmic differences turn off on specific genes in DNA. So even though all of our cells have the same DNA, there are signals that tell particular cells what to do that make them a little bit different from another cell. Okay, so the daughter cells become different. So as I said, we start off with identical cells that eventually change over time, and that process is called differentiation. Differentiated cells are responsible for all body functions. So before a cell becomes differentiated, it's just like all the other cells. And that's sort of, if you think about it, what stem cells are. So stem cells are undifferentiated cells, which means they are still capable of becoming different types of cells. Once a heart cell becomes a heart cell, it's going to stay a heart cell, in other words. But before it becomes a heart cell, it can be manipulated to become maybe a brain cell or a kidney cell. Okay? So differentiation is where the cells become what their ultimate function will be for the remainder of its life. So ECF versus ICF, extracellular fluid, what is that? So extracellular fluid, if you have a cell, the cell is within a fluid medium, and that would be the extracellular fluid, whatever is outside of the cell. And ECF is also referred to as interstitial fluid. You'll see that a lot more in Biology 202. Interstitial fluid is the same as extracellular fluid, the fluid medium outside of the cell. And the opposite of that, obviously, is intracellular fluid, and that's what? The fluid inside the cell, which makes up the cytosol. Okay, so in a cell, so what are the major parts of a cell? Cell components. First you have the plasma membrane, which is what? Okay, that's what it's made of. But what would you say the plasma membrane is? The cell membrane, what is that? The cell membrane. Yeah, that's what it's made of, but just what is it? Right, it's just the outer structure, the boundary of the cell, the outer boundary of the cell. Okay? So the cell membrane or plasma membrane is just the outer boundary of the cell, and we'll look at some of its functions, and then everything inside of the plasma membrane is the cytoplasm. So the cytoplasm is made up of cytosol. So is everybody clear the difference between cytoplasm and cytosol? So the cytoplasm is just the actual space inside the membrane. The cytosol is the fluid that is in the cytoplasm. Okay, so the fluid material, semi-solid material within the cytoplasm is the cytosol. Organelles, you should know what those are as well. The word organelle means little organ, small organs. So within the cell, there are structures that we'll talk quite a bit about in this chapter. And they're referred to as little organs, such as, like I said, the mitochondria, ribosomes, Golgi. Those would be called organelles. And they each have a specific function for that cell. Okay, let's see here. So first let's look at the plasma membrane. So what do we say the plasma membrane is? Just the boundary of the cell, right? The outer boundary of the cell is called the plasma membrane. And so functionally, what's the function of the plasma membrane? So it separates the outside of the cell from the inside, and it serves as the boundary for the cell. But what important function it has is it controls what comes in and out of the cell. Okay, so that's basically it. Plasma membrane function controls what enters and exits the cell. So not everything that wants to get in the cell can get in. And not everything that wants to go out can get out. And that's because the membrane is said to be, what's that word from 103? The membrane is said to be blank because it controls what enters and exits the cell. Semi-permeable, right. So the plasma membrane is semi-permeable because it controls what enters and exits the cell. Okay, so that's basically it for the plasma membrane. What about the cell wall? Where is that? Inside of plants, right? So animal cells don't have a cell wall. So do our cells have a cell wall? No. Okay, so only plant cells and some bacteria. All right, so plasma membrane controls what enters and exits, separates intracellular fluid from extracellular fluid. So it's the boundary between the ECF and ICF. It participates in cellular activities, as we'll see, and then it has a bilayer structure. So make sure you know all this information on this slide, definitely understand it. And the best way to do that is have a picture of it in your mind. Okay, so looking at this picture, where's the ECF and where's the ICF? What color is the ECF? Okay, so the ECF is purple, blue on my screen. And the ICF, it's different up here. It's pink up here. On my computer, it's orange on the screen. But either way, so you're looking at the plasma membrane, and look how it's formed. What's the major component that you find in the plasma membrane? In other words, what does it look like the plasma membrane is made of? So you see a sea of, look like oranges, right? That sea of oranges is what? What part? Right, so remember, you all remember phospholipids? So all of our cell membranes are made of phospholipids. Okay, and the phospholipids are arranged in two layers. That's why it's called phospholipid bilayer. You see the layer on the inside and the outside. So the oranges are there, and the oranges are the head of the phosphate, the phospholipid. So the phosphate group is what you're seeing there in orange. And then the oranges have two little sticks. And those are the what? Those are the tails of the phospholipid. Okay? So phospholipids. So on the test, if I ask you what is the major component of the cell membrane, it is what? Those phospholipids. And do you all remember the phospholipid head is what? Hydrophilic or hydrophobic? So the head is philic, hydrophilic, interacts well with water. The tails of the phospholipid are hydrophobic, and that's because they're fatty acids. So they're the lipid part of the phospholipid. And remember, lipids don't interact well with water, right? Lipids, fats, oils don't mix with water. Okay. Now, what else do we see in the picture of the membrane, the plasma membrane? Right, so you see a lot of big chunks, big blue chunks. So those big blue chunks are proteins. So in the plasma membrane, the major component is phospholipids, the phospholipids. But embedded within the phospholipids are proteins. And keep in mind, these all have a function. So the main function of the phospholipid bilayer is to the semi-permeability of the cell, right? So that's the part that keeps some things from coming in and out. The proteins, we'll see, have a bunch of functions. One of those is to allow some things in and not others. So you have the proteins there, the phospholipids. What else do you see? What are those?
Cholesterol.
Well, good. So you see cholesterol. So cholesterol is another important component of the cell membrane, the plasma membrane. And it helps to make the plasma membrane semi-permeable also. And see, there's one more thing you can see just outside of the cell. Something besides the proteins. What are those kind of, I guess, not polypeptide, but they kind of look that way. Carbohydrates. Okay? So you have those chains of carbohydrates. And so glyco, wherever you see glyco means what? Carb. Okay, so anytime you see glyco, think carbohydrate. So there we have it. So the cell membrane, remember the molecules of life? What are the molecules of life? Good. Not so good. I only heard about 2 or 3 of them. But carbs, lipids, proteins, nucleic acids. So 3 of those we have that make up our membrane, right? So we said the reason they're called molecules of life is what? That's why they're organic. But why are they called molecules of life? Y'all don't remember that? Right. Because all living things are made of carbs, lipids, proteins. And if all living things are made of cells, and all the membranes are made of phospholipid, protein, carb, the nucleic acids are inside in the DNA. So that's what our cells are made of. And you can see it right here in the plasma membrane. Okay. And so those are the components. So the major component of the plasma membrane is the phospholipid. Makes up 75% of the lipids, of the membrane. 5% is glycolipids. What do you think a glycolipid is? So it has a carbohydrate part attached to a lipid part. Okay? And if you see something called a glycolipid, you can see the purple part is connected to the fatty acid. And then cholesterol, then there are glycoproteins, that's a carbohydrate attached to a protein. And then cholesterol, what type of molecule is cholesterol? Carb, lipid, protein, nucleic acid. It's a lipid, so cholesterol is a lipid. Okay, so those are the lipids of the plasma membrane. And the proteins. You can take a whole course on the functions that the proteins serve in the plasma membrane. Some are receptors. You heard what receptors do, right? From chapter 1. So receptors respond to change, stimuli. So we have cells that have receptors. Some of the proteins that are in the membrane serve as enzymes, speeding up chemical reactions. Some are channels. So remember we said some substances like sodium and potassium have a hard time getting through the phospholipid bilayer. So in order for them to move in and out, they have their own specialized doors that open to allow sodium to come in when it needs to, and potassium to go out. So that's what the ion channels are. So those are proteins, and they're remarkable, as you'll see mostly in AMP2, what they do in order to allow our nervous system to work. Okay, so those gates. Then you have cell identity markers, which are proteins that have carbohydrates attached to them. And those, you all heard those before, cell identity markers on the surface of our cells. And that is what allows our immune system to detect what belongs. Okay, so on my cells, my identity markers are going to be different from your cells. So if your cells are introduced into my system, my immune system is going to attack your cells because the identity markers don't match what mine are supposed to be. So they allow the immune system to detect or distinguish from the body cells, foreign cells. And then you have proteins that serve as adhesion molecules connecting two cells together. Okay, so the glycocalyx, the main thing to know about the glycocalyx, what does that glyco mean again? Carbs. So in reference to the cell, where do we find these carbs? What did we just say? Looking back at so they're the purple things here. So the purple structures outside of their cell membrane are carbs, and together they combine to form something called the glycocalyx. Okay? And the glycocalyx is what allows our immune system to distinguish self from foreign. Okay, and those are just some of the different functions of the glycocalyx, also referred to as sugar coating. Sugar coating. Our cells are sugar coated, and that would be the glycocalyx. Some of the organelles on the surface, most of our organelles are inside the cytoplasm. These are referred to as surface organelles because they project on the outside of the plasma membrane. Cilia and flagella. So this picture shows you cilia. You all remember where cilia is located in our bodies? Okay, so in the airway, the respiratory tract, and they have specific functions. They kind of help to move debris up to our throat, so that if you imagine dust getting into your airway, instead of allowing the dust to get all the way to the lungs, where infection may set up, the cilia pushes that in mucus, and actually those cells, the orange cells around, are goblet cells, and they produce mucus that covers. It's actually on top of the cilia. So the dust particles get trapped in the mucus, and the cilia push the mucus up to your throat. So when you clear your throat like that, you're getting rid of that debris from your respiratory tract. All right, cilia and cystic fibrosis. So again, this is another picture showing you how the cilia actually work. Okay, so the cilia are there for a purpose, to push the mucus layer. You can see the mucus layer up top. And the cilia is sort of bathed in a saline, watery fluid. And cystic fibrosis, you all heard of that? What happens with cystic fibrosis is it's a hereditary disease in which cells make chloride pumps but fail to install them in the plasma membrane. So the chloride pumps help to secrete that saline layer. So the saline layer is important. So remember, I said the saline layer here is watery, so that allows the cilia to bend. If the saline layer is not there, the mucus sits on top of the cilia, and the cilia are not able to move. Okay, so that saline layer is important. And what happens is without it, thick mucus plugs occur, and they build up in the pancreatic ducts and respiratory tract. So you can imagine a person who has cystic fibrosis, they're probably going to have a lot of lung infections because there's nothing pushing that debris back up to the throat. It keeps building up in the lungs, setting up infections. And you have also the pancreas is affected because of the ducts there being plugged by these mucus plugs. Okay, and so that's cystic fibrosis. And then flagella. Which cells in our body have flagella? In the human body, I should say. So only sperm cells, right? In humans, sperm cells are the only cells that have flagella. The cytoplasm, so we mentioned the cytoplasm. All right, so the cytoplasm, now we'll get into the organelles within the cell. So first of all, the cytosol is mainly water, mostly water. So most of the water that make up our body is found inside of our cells. Also within the cytoplasm, you have proteins, salt, sugars, electrolytes. Cytoplasmic organelles, again, are the little machines within the cell. And inclusions include glycogen, lipid droplets, crystals, vacuoles, just other substances not considered organelles that are in the cytoplasm. Okay, so the nucleus, I guess so we'll go through the function of each organelle here. And you should know a little bit about each one, the structure of it, that is, as well as the functions. So the nucleus, if I asked you a function, you basically just say what? DNA and RNA storage. Good. Okay, so that's where our genetic information is stored, in the nucleus. Okay, so the nucleus also contains chromatin and something called a nucleolus. Okay, so the nucleolus and the DNA material, which is the chromatin. And the nucleus also has pores. So what do you think the pores are there for? Somebody remembers, I'm sure, from 103. What's the role of those big pores in the nucleus? Very good. Good. So RNA that starts in the nucleus comes out to the cytoplasm through the nuclear pores. So they're just pores. And by the way, the nucleus has a phospholipid covering as well. And those pores allow RNA to escape the nucleus at the correct time. DNA, however, DNA being double-stranded is not small enough to go through the pores. So DNA is always contained within the nucleus, whereas RNA can come out of the nucleus into the cytoplasm. All right. The cell's nucleus, function, information storage. That's where our DNA is located. DNA, instructions for every protein in the body. Gene, DNA instructions for one protein. So sometimes it's still hard for me to make sense of what we know as DNA. DNA is what makes us us, right? It makes us unique. But our DNA is the same pretty much in each of us. So what is it that DNA is actually doing? What is it that DNA actually does? So it's right. So DNA is just a code, right? And it's a code that determines what proteins can be made, as we'll see. So the genetic code is the chemical language of DNA instructions. So it's just a long sequence of instructions that allows the cell to make a particular protein, whatever protein it needs to make at a particular time. And you all should remember the sequence of bases, or the types of bases: adenine, thymine, cytosine, guanine. Those are the nucleotides that are found in DNA. And the sequence of those nucleotides will code for a particular amino acid, as we'll see later. But all it does, as we said, is tells the cell which proteins to make. That's it. Okay? Triplet code, 3 bases equals 1 amino acid. 3 bases equals 1 amino acid. RNA structure compared to DNA. And these are some things you all should know. In RNA, both RNA and DNA are made of a sugar and a phosphate group and a nitrogenous base. The sugar in RNA is ribose. DNA is deoxyribose. So that's where the R and the D come from. Which sugars do they have? Number 2, their difference is also in the nucleotide, or the bases within the nucleotide. Adenine, guanine, uracil, cytosine. So what's the difference between that and DNA? Right, so there's only one difference in bases between RNA and DNA. So in DNA you have thymine, in RNA you have uracil. So remember that on the test. What I may do is give you a sequence and ask you what would be the corresponding RNA sequence based on that DNA sequence. RNA is single-stranded and does not have a helix, whereas DNA is double-stranded and has a double helix. Okay, the DNA molecular structure. So again, the nucleotide is made up of sugar here, which is deoxyribose, a base here, which is adenine, and a phosphate group. Okay, so that picture there is showing you the adenosine nucleotide. And there's the double helix. So you have 2 strands of nucleotides, and what you here have called the complementary base pairing. You should know which nucleotide is always across from a particular one. So if you have a guanine on one strand, what's always opposite or combined with it? Cytosine. Okay, so you should know which bases are complementary shared in the DNA double strand. So thymine is always forming a bond with what? Adenine. And that is in DNA. So you see the A's and T's form a bond, and then the G's and C's, guanine and cytosines. Okay, law of complementary base pairing. One strand determines the base sequence of another. How a gene works. So first of all, what did we say a gene is? I think we did define that before. DNA instructions for one protein. Does that make sense to anyone? So if you have a sequence of nucleotides, and remember nucleotides are thousands of bases long, the DNA molecule. So if you have one strand, A, T, A, G, C, C, T, A, C, C, C. That's one DNA strand. So what will be the complementary strand? T, A, T, C, G, A, T, G. It's a C. Oh, G. Okay, so that one strand is going to complement the opposite strand. Now, so what a gene is, what is a gene again? Right, so it's the sequence of this particular sequence of DNA that codes for one protein. And again, these are usually thousands of base pairs or nucleotides in length. But let's just say this part is going to code for a protein. A protein like what? Can you think of a protein in a cell? And think of it in real life terms. Any of our cells have to make proteins. I think we mentioned one, something found inside of red blood cells, hemoglobin. So hemoglobin is a protein, right? Our red blood cells must have hemoglobin to transport oxygen. So red blood cells have to be able to make hemoglobin, and this is how it does it. But of course, it would be more like thousands of base pairs, but this would be called a gene because just that segment is going to make hemoglobin. Further down, there may be another segment that makes melanin, or you know what I'm saying? So any other thing that the cell may need, that's what the DNA is set up to do. So, but before it does that, before it codes for a protein, remember, one of these strands is going to be read and interpreted in order to make an RNA strand. So you start off with DNA, and something happens here, and you end up with an RNA strand. And what is this called? In order for the DNA strand to make a copy of RNA, transcription. Good. So transcription is where RNA interprets DNA in order to make its own strand. Okay, so what would that RNA strand look like? So we have RNA here, and it's going to be based off of this strand of DNA. So what would the RNA strand look like? So you have U. No, it's actually what's wrong with that? So we have T, and across from T, in DNA you have adenine, right? So across from the T here, you have the same thing. I just can't find my Okay, so basically what you're doing is building a strand based on this, but you still have complementary base pairing. So across from T, you always have what? A. Okay, across from A, you always have T in DNA, but there is no T in RNA. So it's going to be U. Okay? So this is my RNA strand. AUA, G, C, C, U, or T. Yeah, T. Okay. Yeah, this is an A. I was on this one. T. C. Okay, so this is the RNA strand that codes from that DNA strand. Okay, now, then the next step, so based on this RNA, so this is going to be the mRNA, right? Remember the types of RNA? mRNA, tRNA, and rRNA. Good. All right, so all of this. So you have 3 nucleotides that code for what? Amino acids, right. So based on this RNA strand, it says here the RNA interprets DNA code, uses those instructions for protein synthesis. So protein synthesis, what is that again? Building, right? Synthesis is building. So building a protein. How do you build a protein? You need what? Amino acids. So every 3 nucleotides codes for 1 amino acid. Yes. Okay, I was hoping to see the actual amino acids, but that's basically what these are. So this is 1 amino acid. That's the second amino acid. This is the tRNA. So if you remember, the tRNA brings in the amino acid, okay, based on what this code is. So it's like an assembly line, pretty much. And it's real intricate. It takes really a couple of days to go over DNA, so I'm just giving you all the overview. So remember that the mRNA leaves the nucleus, and once it gets into the cytoplasm, you have what's called translation. So transcription is the coding of RNA from DNA, transcription. And then translation is like a difference in language. So if you're translating one language from another, you have the language of nucleotides translated to the sequence of amino acids. So there's the translation. The amino acid sequence of a protein is ultimately determined by what? The nucleotide sequence of the original DNA. Okay? Because if you didn't have TAT, you wouldn't have AUA. And AUA codes for a particular amino acid in the protein. All right. Let's take a break here.